everyone at this ESID Grand Round webinar called The Power of Flow-Based Assays in Diagnostics of Inborn Errors of Immunity. I welcome you also on behalf of Klaus Warnatz and Clara Franco Jarava, the co-chairs, and also on behalf of the speakers. We are delighted to have a session dedicated on flow cytometry after an interesting discussion about genetics in the previous Grand Round. So what I would like to start off with the question, do we still need flow cytometry in the era of NGS for diagnostics of inborn errors of immunity? And I think definitely yes. Because flow cytometry plays a role in different steps of the diagnostics for inborn errors of immunity, from the clinical evaluation, the pre-genetic workup, but also during the confirmation and also during the clinical management and treatment procedure. So that's also what we would like to discuss this afternoon. So with the Euroflow Consortium on PID, it's a European consortium, and we developed over the years several tubes that allow the analysis of the lymphocyte subsets, especially in patients with inborn errors of immunity. And we compose it as like an orientation or a screening tube. And what is displayed here, the composition and also the markers we used. And you can define several B and T cell subsets. And that's as a basis for screening of inborn errors of immunity, it can be a great advantage. And you can define the different populations. And I think the strength of what we did with the Euroflow Consortium is that we also generated a lot of reference values, because when you use flow cytometry in diagnostics for inborn errors of immunity, it's crucial to have the reference values of the age-matched controls. And that's also what we did over the time. And we not only standardized the procedure, but also we validated the performance of this tube in 100 genetically defined inborn errors of immunity. And we think from the Euroflow Consortium, that's really the power, that it's standardized and you have the reference value and it's validated. So that allows you to apply it in various laboratories, and that we can perform multi-center studies. And this is also an example of what we did. You can not only measure the amount of lymphocyte subset, but it also allows you to give some pattern recognition that different inborn errors of immunity have different profiles of the lymphocyte subset. And that gives you a different view on the analysis of lymphocyte subset, and especially in the context of the diagnostics. So on top of this PID orientation tube, you can further build on that with several other tubes focusing on SCID or T cell subsets or isotypes or other B cell subsets. I will not go into this today. So for today, we invited 4 excellent speakers, and they will focus on different aspects of flow cytometry and flow-based assays in the diagnostics for inborn errors of immunity. The first speaker is Tomas Kalina. He's professor at the Pediatric Hematology and Oncology Department at the Charles University in Prague, in Czechoslovakia. And he's board member of the Euroflow Consortium, and also of the CD Nomenclature Committee member, and he's also very active at ISAC. He will talk about the sense of T cell phenotyping in diagnostics of inborn errors of immunity. For this Grand Round webinar, he will give his presentation, and we can have some questions after his presentation. And for the other speakers, we think it's best that they will present after each other, and that at the end we have panel discussions for which you can ask questions also in the chat. And also then we have a discussion for all the topics together. Because I think especially for flow cytometry, we really need each other in the field of inborn errors of immunity. And I think we will hear interesting presentation, and we also hear more about challenges we need to address. And also there will be examples that that look forward to further collaboration within our society. Now I'm happy to give the floor to Tomas Kalina.
Thank you very much, Mirjam, for your kind introduction. I hope that you can see my screen now. So I would like to talk about the role of flow cytometry in the world of inborn errors of immunity. I think we all agree that primary immunodeficiency is a beautiful but challenging diagnostic problem that we have to approach with several modalities. We know that clinical presentation is definitely important. Gene mutation by any genetic means is the key. And still, I think that there is the important point is that we have to put together all these modalities to come to the right diagnosis. What is a bit challenging is also that the spectrum of disease severity is actually very variable from emergency cases like SCID, where we need to make the diagnosis as soon as possible and proceed to the transplantation that is life-saving, to other milder immunodeficiencies where the diagnostic procedure is not needed to be in a matter of days, and the patient has a variable and sometimes not that severe presentation. I think my view on the role of the phenotyping in PID is definitely twofold, and the key here is the diagnostic. So there is a need for some diagnostic contribution, and I think that for especially the severe immunodeficiency, the SCID and CID, and for some specific disease like ALPS, this is clearly supporting modality that can support the diagnosis making. It's not it should never be used alone, but it's still supporting. And then there are multiple other immunodeficiencies where we know the gene mutation, where we want to see that there is a correlating phenotype, but the phenotype itself is not clearly identifying the gene mutation. It's only correlating. And then there is a role for flow cytometry in other specific types of assays. For instance, in newborn screening, it's increasingly used after the initial screening of the track and cracks. The confirmation is done by flow cytometry in some countries, like in ours. And there are a few, especially few countries where it is worthwhile to do identification of a missing protein, especially if there is a high prevalence of that immunodeficiency in a given country. And as an example, I think you can show it with a perforin deficiency, class 1 or class 2 deficiency, or leukocyte adhesion deficiency. And there will be a specific talk on this, that flow cytometry is used as a function readout of altered function. So in the most severe cases like SCID and CID, in comparison to healthy, you see a clear absence of T cells. This is easy, and this is where flow cytometry was always very good in making very fast assessment of absence of T cells. So this is still true. However, in basically up to 1/3 of all cases, you would see some T cells present even in the patients with a confirmed mutation leading to SCID, and this can be due to maternal engraftment, like in this case, or it can be also in cases of oligoclonal expansion, in cases of hypomorphic mutations, or Omenn syndrome or leaky SCID. So what you see is that either there is a complete absence of T cells, or there is a presence of T cells, but there is a malfunction or non-function of the T cells, or there is even some type of dysregulation caused by the T cells. So our goal is to find the absence of T cells and possibly find the clear abnormality of T cells that are present, if there are any. The typical findings in SCID or CID, if there are some T cells present, are that they lack the naive forms, because all the T cells present are generated by peripheral expansion. They are activated because there is the expansion going on, and they also lack the more a finer population of naive T cells that are called recent thymic emigrants, which is a sign of a absent thymic output. In healthy newborn, you can see that there is a majority of T cells. I need to switch on my pointer as a laser pointer. So you see there is most of the T cells present are naive T cells. There are very few, if any, activated, and large part, about 2/3 of T cells are naive T cells are recent thymic emigrants. So you see, these should be here, these should be absent, these should be also here in a normal, healthy newborn. As Mirjam mentioned, we are using the Euroflow approach with two types of tubes. One is the PIDO, the orientation tube, and one is the focus tube on a SCID RTE, detection of SCID patients and detection of recent thymic emigrants. So you see that the proper gating follows this logic where you only select non-activated cells without RO expression in this patient, although there is a strong presence of T cells that are of maternal origin that we later on confirmed by molecular genetics. We see there is nothing in this region of the non-activated naive T cells, and consequently there are only few dots of recent thymic emigrants. So clearly there are T cells, but they are not naive, and they are not they do not contain recent thymic emigrants. And what you can also see is that a large portion, actually majority of them, is activated by HLA-DR positivity. So Mirjam has already introduced you briefly into the scheme, which I will not go into the detail. I would refer you to the papers that you can look up. There are several of them, but you can look up on PubMed by typing Euroflow and Burg. And I will only focus on the orientation tube and the SCID RTE tube. So the orientation tube is basically the classical TB-NK. This is now highlighted in red. And then in orange, you see the added markers. So these two markers are giving us the chance to look at the naive forms. This is what gives us a chance to look at the subset of B cells together with CD27. And the SCID RTE tube contains the other isoform, the 45RO, to kind of make it complementary with the first tube. And then there is markers that still allow us to identify naive cells, CD62L and 45RO, and then the recent thymic emigrants positive for CD31, activation by TCR, by HLA-DR, and we can also check whether the T cells are of TCR gamma delta, or if they are gamma delta negative, then they should be alpha beta positive. Now I can jump to the evaluation of our cohort of patients up to 2 years of age, where we could see that in the controls, which were patients shortly in the first weeks after birth, you see that absolute counts are clearly higher in the normals. You see that in the patients with SCID or other severe combined immunodeficiency, which are listed here to the right, the individual mutations, we see that mostly the T cells are decreased. But what is important is that when we look at the most clear marker that allows us to make a diagnosis, you see that if you look at the activated DR positive cells, and we also look at the RT and naive, then we can achieve basically with this flow assay 100% sensitivity. So no patient with SCID would actually escape the detection. And with the other PID that are listed here, you see there are multiple, but there is also one patient with ALPS that would basically not be easily detected by just looking into the naive recent thymic emigrants and activation. So this is why the sensitivity would be lower. And if we compare these two tubes, we gauge the naive T cells here as a RA positive and 27 positive, and here as a 62L positive and RO negative. then we see that in classical SCID, there is nothing to be seen because this patient had no T cells, so this is easy. And then the patient that I showed you as an example lacks naive T cells by both methods and still shows a profound activation and lack of RTEs. If we look whether the PIDOT tube is actually good enough as a initial orientation or screening for SCIDs, we see there is a very high degree of correlation in the number of naive T cells in patients with SCID, other PID, and other PID. So basically, you can use the PIDOT tube as an initial evaluation, and then for confirmation, you can use the SCID RTE tube that gives you better precision, especially for the other patients with the other PIDs. But the PIDOT will not miss a SCID. Then you may ask, why do we actually need it? So I am convinced that for this very crucial diagnostic issue, you need to investigate two tubes before you make the diagnosis, because it's in the interest of the patient to be properly diagnosed. So yes, it's twofold more expensive than it needs to be, but I still believe this is meaningful. And then my last stop is at the ALPS. So ALPS by cytometry, you know that we need to look at TCR alpha beta T cells only, and then see if there is an expansion of double negative T cells that are typically hallmark of ALPS. However, the diagnostic problem with this simple measurement is that there is many patients with ALPS that are around the boundary that is drawn as a normal versus abnormal number of double negatives. So recently there was a beautiful paper by the Freiburg group showing the different numbers of double negative T cells in ALPS, and showing that not only you have different numbers, higher numbers in ALPS, but there is also a few other mutations like STAT3 gain of function that will present with increased double negatives and CTLA4. But if you add what they called ALPS signature, which was discovered by broad immunophenotyping using CyTOF, you see that in comparison to healthy donors and gain of STAT3 gain of function and CTLA4 deficiency, the ALPS patients have many cells in this ALPS signature region when you focus on double negatives. And if you want to deconvolute what it is basically a high expression of CD38 and 45RA. So this is how ALPS patient look like when you look at the double negative T cells by these two markers. This is a second patient, and you see that that there is always a great majority of the double negative T cells is presenting with this ALPS signature. And if you look at the numbers, the ALPS signature is really specific for the ALPS patients, and this is much lower for the other two types of mutations that present with increased double negative T cells. So if you want to ask about our patient that I showed you on the first slide, yes, this patient did have the ALPS signature cells in a 66%, so even the 7% of double negatives is already high enough to make a high suspicion for ALPS, but this is definitely reassuring, and it was then genetically confirmed. So ALPS is now much better diagnosed with a ALPS, so-called ALPS signature. And on my last slide, I want to just show that in the Euroflow data set that Mirjam has mentioned, you can somewhere deep in the supplementary, you can find a big table that shows you individual groups of mutations and how frequently there is an abnormality in terms of decrease or increase of the individual subsets. And you see that basically the naive CD8 cells, the naive CD4 cells, and switched memory B cells are among the most frequent cell subsets that you need to look for if you want to find abnormality in many of the groups of immunodeficiency here grouped by the IUIS tables. So with that, I want to summarize that in the world of NGS, we still want to make a diagnosis by confirming the missing population in SCIDs, because that's the transplantation is the next measure we will take with these patients. So we want to confirm this beyond any doubt. We can still use the Euroflow tubes to confirm the criteria of CID, even in the absence of genetic diagnosis. And then we can use, beyond diagnostics, we want to use flow cytometry to better understand the biology, to describe in more detail the new PIDs that are discovered. We want to use it to monitor the targeted therapy effects. And in some cases, we want to directly detect missing protein. But this is challenging because you have to have the assay in stock, and it's very rare that you find a patient here. And then I did not talk about functional tests, but they will be mentioned later. And I think the first two steps should be easy to set up in any lab that should be ready to diagnose a patient with SCID. And the later tests are only meant for specialized laboratories because they require more expertise, more elaborate SOPs and reagent portfolio. With that, I want to thank to my colleague from my university and also my colleague from the Euroflow Consortium, and thank you for your attention.
Thank you very much, Tomas. And as Tomas will have to leave us, really please ask questions now because he will not be available for discussion later. So thank you very much for this very nice introduction into the use of flow cytometry, and especially in the context of T-cell deficiencies. One of the first questions which popped up was a question in regard to your last topic, in regard to ALPS. And the question was whether this signature population is also helpful in other forms of ALPS, so specifically for fast ligand deficiency, for example.
So, so far, I have to say that the data from Freiburg, as well as our few patients that we diagnosed and many more that were not confirmed as ALPS, actually show that we did not find any patient that would kind of either fall out being fast and not being ALPS and not being detected, or the other way around, having the ALPS signature and not being confirmed as ALPS. So far, it seems that in all forms of ALPS, we can use this ALPS signature.
Yeah, right. Thank you very much. And I think the data is still pending to really prove that formally, but I think that is going that direction. I think the beauty of this, in my eyes, is that that it proves the idea of indicator populations, and very nicely shown by Maria Elena and all the people, that you have a large screening panel, like a CyTOF panel or whatever your large screening panel is, and identify new. So do you see other populations coming there? And iNKT, so and used NKT cell invariant NKT cells have been postulated for some of them, right? And there are others. So do you see other T cell populations where you see, yes, they might fulfill similar criteria, like mite cells have been screened, but I think they didn't quite make it so far as I can tell.
Yeah, that's I think that I'm for me, this is again, as you say, a beautiful example of this approach. But to my frustration, I have not seen a second population that would have such a unique and in the same time specific and sensitive properties. So yes, there are hopes for several, but I have not seen this really proven to this extent as it is in ALPS. But I think that perhaps you will learn more through the other talks. And I think my hope for the next index populations will probably be more linked to the functional profiling of particular subsets rather than just the immunophenotyping of the subsets, which is a bit more elaborate, but we'll see. Maybe there will be some more index populations, but maybe the ultimate contribution of flow will be to actually look at particular cell subset and see the function that is altered.
Next question is on treatment effects on your readouts. So there's a specific question, impact of steroids, sirolimus on the double negative signature in ALPS.
That I cannot respond formally. We have not looked into it, I believe. So I don't know.
Yeah, so I think Stefan has looked at it, and I hope he's not on there because I don't remember quite well either, but I should, hearing him all the time. But I do think so. Sirolimus can clearly decrease the double negative counts and therefore will affect it. For steroids, I'm not aware that it does so much. So I think that is as possible. Yeah, right. And the last one, is ALPS signature useful for diagnosing ALPS-like diseases such LRBA or useful differential diagnosis? So the question is again, yeah, so you have increased double negatives, right? And the question is again for the signature. And yeah, Tomas.
Here, I would say that when you do see an increase of double negatives, TCR alpha beta positive, and there is not an increase of the ALPS signature population, it is more likely that it's one of the other gene mutations that you mentioned. So it's at the moment, it seems to be specific for ALPS, and by exclusion, you can make a conclusion that it is not the other diseases, which is probably good for the management of the patients, because this
Absolutely.
would be somewhat different for the other forms of EIA.
Right, you're absolutely right. And I think that's a big concern of Stefan's, to understand what he now calls ALPIT, so to enlarge it. And they are having double negatives, but not with that signature. I think it's probably quite fair if you have more than 10% of double negatives, truly TCR alpha beta positive, but double negatives, ALPS becomes very likely. I think the others can have up to 10%. They can have more, but it's rare. It's much more rare than in ALPS. Yeah. Thank you very much. And then Adam asks a question: is the information available, screening parameters around infections and effect of treatments? That's what we just tried to address a little bit, because that obviously confounds findings, and I totally agree.
Yeah.
So the answer is, I guess we don't have the full range of infections. They're all different kinds of.
I mean, in general. So in general, I don't think we have a clear signs of infection in those readouts, and I think it's still the gray zone where we need to realize whether this is if the abnormality that is not as severe as in SCID, whether this is caused by reactive situation based on some infection, or whether this is PID is definitely a question. And what is little discussed, but I think we should pay more attention to it, is watching these patients in time, because what we see in many diseases, if it's a gene mutation caused, then the phenotype is fixed and it's not changing much, at least for some of the major populations. And in a different reactivity to infection, you will see this will change a lot regarding based on in which phase of treatment the patient is.
Yeah,
so time might be the parameter that we need to look into more.
Yeah, no, thank you too much. I think just in regard of times, there's one last question: whether is it really important to have the mesenchymic emigrants in patients with SCID, or do you actually get around with the naive counts and you don't need really the mesenchymic emigrants?
I can understand if some people decide not to have mesenchymic emigrants, but so far, I think if I value against the severity of the decision against the measurement of one extra population, I'm in favor of including them, because it makes the diagnosis much more firm. And I think you should not also forget that in the lab that is bound to diagnose SCID, you can end up with a patient which will not be ultimately diagnosed by NGS. So there will be a patient that doesn't have his mutation found and still might need a transplant. And in that case, you need to fulfill the ESID criteria for CID, which do contain the mesenchymic emigrants. So I think even from this perspective, you want to have that test available, and I think it's worthwhile using it. Although I can understand that the information can be redundant if you have the naive and activation phenotype as well.
Right. No, so thank you so much. Just in regard of times, to give also the other speakers the time and the discussion. Thank you so much, Tomas, for this excellent start into this. And I think please ask more questions, and we will try to forward these questions to the speakers later so that you will have an online response. So I see there are a few more popping up. The question I was also interested in, absolute counts versus relative counts and all these things. So I think there are quite a few things stimulated by your talk. So thank you so much, Tomas, and all the best for now.
Thank you.
So now it's my great pleasure to introduce Samuel Chiang. Samuel is an assistant professor and assistant director for development, and he is heading in the diagnostic immunology laboratories of the Cincinnati Children's Hospital. And he has a very strong interest in developing diagnostic assays in the field of inborn errors of immunity. So thank you very much, Sam, for joining us and presenting us your thoughts on NK cells.
There we go. All right. So good evening, good morning, good afternoon, good night, wherever you are in the world. Hello. My name is Sam. Thank you to the organizers for inviting me. I'm currently at Cincinnati Children's Hospital, North America, where I help to develop and to validate new clinical tests with focus on IEIs, specifically using flow cytometry. But before that, before this, I was in Sweden for 9 years at Karolinska. So I'm going to share some data on that. I'm going to try to do 3 things today. Number 1, talk about functional NK cell testing, and this is the degranulation test. This is not the chromium release NK killing. This is the CD107A test. Number 2, a little bit on Treg function, where we try to come up with an easy, simple test for CTLA4 and LRBA. And finally, I have 1 slide where I look into my crystal ball to see where we are all headed in this field. All right, let's get started. I'm not going to introduce HLH, which is hemophagocytic lymphohistiocytosis, primary HLH, FHL, whatever you want to call it. I'm not going to introduce that. Just 1 slide on how we actually run the test. So we always start out with whole blood. This should be a green cap. It's normally heparin. Phycol it, plate it overnight to rest the PBMCs. The next day, you have your PBMCs here, and you stimulate them by adding in K562 or PI, whatever, some kind of stimulation. Leave them to mingle for a couple of hours, and then after that, drop in some antibodies so that you can differentiate out T cells and NK cells. But more importantly, if an antibody in there against CD107A, you stain it, you run it on cytometer, and then you analyze the data. What I'm interested in is to explain what happens here, what happens during this interaction. So we have here a killer lymphocyte, which could be an NK cell, could be a cytotoxic T cell, and these killer lymphocytes would normally identify cancer and infect the cell and say, hey, I'm going to kill you. And when it identifies it, the first thing it does is it forms this immune synapse. And then you see these granules here. And so these granules are filled with perforin and granzyme. But the pink things that are normally on the inside of the granules are CD107A. So in a resting state, CD107A are found in the granules, but not on the surface of these cells. So when an immune synapse is formed, these granules will polarize towards that, and the membranes of the granules would fuse with the membrane of the cell. And that would release the perforin and the granzymes, and the perforin and the granzymes will initiate apoptosis. But the CD107A that was once found on the inside of the granules can now be found on the surface of the cell. So this is our focus, the CD107A. If we see CD107A on the cell surface after the interaction, it means that some of these lytic granules have fused with the cell membrane and perforin and granzyme was released. However, if we do not see that CD107A is on the cell surface after the stimulation, it means that the granules have not fused. And by extrapolation, that means that the target cells have not been affected. So that is the theory of how this test is being carried out. This is the data. This is data that I started collecting while I was a PhD student at Karolinska under Jan Bryson, continues to this day. This is more than 10 years worth of data. On the top row, you will see genes where we know that cytotoxic lymphocyte exocytosis is affected. On the bottom row are IEIs that should not affect cytotoxic lymphocyte exocytosis. There's more than 90 patients here. So at the time when we started, the standard was K562 stimulation. You get PBMCs and you throw in some K562 and you look at CD107A, degranulation or exocytosis. But soon we realized that sometimes it was kind of difficult to differentiate between the normal controls here from the blood bank and these patients. And so we thought to ourselves, hey, we don't want to reinvent the wheel. Can we make this test better? So we tried a whole bunch of stuff, but we finally ended up with two other stimulations. The first is we looked at NK cells after we stimulated with P815 coated with anti-CD16 antibody. And the second test here, the third test, sorry, is looking at T cells after we stimulate them with P815 coated with anti-CD3. And this is the OKT3 clone. So the first point that you can see for these, so we have different genes, right? FHL3, 4, 5, Shigeki-Gashi, and Grishellis. We have 5 different genes. We have these 3 different tests. We have the normal controls up here. This is 200 individuals from the blood bank, and we have all these patients down here. The first point that I'm going to bring across is that all the patients have very low or no exocytosis. So this assay, all 3 of them work really well. But this we sort of know from years ago. This is not news, right? We know that these assays work really well, especially the K562 has been well published. What excites me more is after running so many samples, having this large cohort to compare, we can see that there are better tests that we can run for the diagnosis of FHL rather than just the K562 test. We can stimulate with anti-CD16, we can stimulate with anti-CD3. There are better tests out there. Thirdly, we have normal ranges, and with this compilation, we have what I would call an abnormal range. We now know how abnormal patients that are affected should look like, where they should lie. And to go one step further, we can see that these two new tests have a much better signal to noise ratio compared to the K562, right? You have a lot more space in between. And what I would suggest is that in the future, we could be discovering new deficiencies using this test, where we see instead of no or hardly any function, we see some deficiencies that show partial deficiency, which is partial reduction in function. All right. So as a compilation of that previous slide, those are the first row is here, the exocytosis deficiency, and the second row is here, the other PIDs or IEIs. But what we also collected is a whole bunch of patients where we call it hyperinflammation, MAS, SOJIA, these are patients, sepsis in the ICU. We also have a lot of transport control. These are unrelated controls shipped at the same time. We also have parental controls, father, mother, familial controls, brother, sister, uncle, etc. And I wanted to bring this up because in the real world, we are not testing our patients that we think will have a degranulation deficiency against blood bank controls. We are trying to differentiate these patients from these other patients and other family members that could be affected. And so this is the real comparison here. The next point I want to bring is when this is published as a research paper, we will look for p-values. But in a clinical lab, when we're trying to push this towards a clinical test, we look at accuracy. And very simply, a simple way of understanding this is trying to draw a straight line through the data and trying to use this straight line to differentiate out all the affected patients and all the patients that should not be affected. And we want to see how many false positives we have and how many false negatives we have. The lesser we have of these false results, the better. And when you look at these 3 tests, you can clearly see that there are a significant more number of false positives when we are using the K562 test, meaning that test A and test B here, the ones that we have developed, newly developed, are a lot more accurate compared to the standard K562 test. Right. So to conclude this section, we have lots of samples, we have lots of patients, and we have a healthy range, but we also can create an abnormal range. We also have a transport range. We know how heterozygous carriers would look. We know how other patients that had transport stress would look after 24 hours or 36 hours travel through winter storms or through summer heat. How would they look in the lab? And these are better normal ranges to use compared to just healthy adult controls that we procured from the lab. We didn't reinvent the wheel, we refined it. We refreshed pre-existing tests, and that's what I feel would happen in the future. The standard tests will not go away, but we are just trying to make them 2 steps better or 3 steps better than what we have today. All right. And what the hope is with this better signals, less noise, we get better clinical accuracy. All right. Part 2, shifting gears, we're going to talk about Treg function, and this is in the context of CTLA4 and LRBA deficiency. And I clearly realized that there has been a very good grand rounds in February 2023. You can view that online. It's excellent. And I'm not trying to replace that. And what I want to do is to share Cincinnati's experience with this. It all started when we were trying to come up with a very simple LRBA phenotyping test to detect LRBA patients. Everybody is familiar that Sigma has this rabbit polyclonal antibody. So we got that, and we got in a bunch of LRBA patients, a bunch of CTLA4 patients, a bunch of controls, and we detected 1 patient out of all of that. But a couple of years ago, Sigma, the same company, also came out with a mouse monoclonal antibody. And this has better performance. We see that for the LRBA patients, we get to see more, it detects more of these patients. And all of these are all deletions and truncations down here, whereas all the missense mutations are all up here. From this data, we immediately see that no, this isn't going to cut it. Like more than 50% of our patients will not be detected. LRBA patients will not be detected. We need something better. Instead of a phenotyping test, we need a functional test. We know that there's a transendocytosis test. It's great, but you need really good hands. You need the special cell line, which is not commercially available. It's not open to us. So we read all of Bodo's Greenbacker's paper and David Sands' paper, and compiling all of that, we came up with the simplest test that we could. And this is how it works. Again, we start out with whole blood. We isolate T cells. We stimulate the T cells with anti-CD3, CD28 beads overnight. The next day, we stain them with antibodies, various antibodies on the surface and intracellularly, run them on the cytometer, analyze the data. Good. This is how the data looks. We have CD4 here, and we have dead cell marker, just so that we can get out live CD4 cells. This is how we define Tregs, CD25, double positive with FOXP3. So these are Tregs, and we have mock stimulated PBS. We have stimulated, that's the CD3, CD28 beads. This is how the data readout looks like in a healthy individual. We have CTLA4 staining, and we stain it twice. We first stain it on the surface, and then we fix and perm, and then we stain it again with CTLA4, same clone, different color. So you see both CTLA4 in and out of the cell. When the cells are not stimulated, you have some CTLA4 in the cell. You have no CTLA4 in the cell, on the surface of the cell, sorry. But after you stimulate, you see that there's a lot of CTLA4 in the cell, and this gets pushed to the surface. We have lots of CTLA4 on the surface. How does this look in our patients? So again, a control here and an affected patient here, you see that most of the Tregs would maintain very high levels of negative levels of CTLA4 in the cell, as well as on the surface of the cell. Now, this gating of low, medium, and high was kind of challenging, and different technicians running this would gate it slightly differently. So we decided that, hey, you know what, we're just going to take the entire population and look for the median fluorescence intensity. So that's going to be our readout. The median fluorescence intensity of the entire stimulated population, that's the population in red. This is what we found. We have LRBA missense, LRBA deletion and truncation, CTLA4 deficiency, and these are healthy controls from the lab. So these are adult controls. This is the ratio of the intracellular CTLA4 when comparing patient to shipping controls. MBFI, CTLA4 in the cell. And you can see we can draw a very nice line throughout here with this kind of accuracy. The same thing for the CTLA4 on the surface of the cell. So they both work in the cell as well as surface expression of CTLA4. We are not completely satisfied with this. We think the controls can be bumped up a little bit. So we are refining this test now, but this is how the levels, the data looks. So we are confident that we have a working model, proof of concept that this test has potential to be developed in a clinical lab. My final slide is what the future holds for IEI testing in a research or clinical lab. I have 3 points here. More and more, we are moving towards functional testing. As I showed you in a previous example, sometimes the phenotyping test just doesn't cut it. Antibodies have their limitations. Sometimes polyclonal antibodies would drift with time, the specificity. So I warn you against that. When if you have a polyclonal antibody, be very careful. But we see that more and more we are interested in the function of a particular cell type. But that doesn't mean that phenotyping tests will go away. For like SCID, for screening for perforin or SAT, or all these many other diseases, the phenotyping tests will not go away. It's great, it works, and it will stay for a long time. But more and more, when we're developing new tests, we are trying to develop functional tests. We are having new methods. We have better machines, we have better kits, we have better antibodies, we have better transportation. And all this will lead to better results in the end, and new tests that we can develop. Finally, RNA signatures. We know that DNA makes RNA and RNA makes protein. A month ago, we had the grand round where we talked about genetic sequencing. Today, most of our talks are dealing with the protein end of the spectrum. But where I see a hole in the IEI diagnostics is looking at the RNA and how the signatures of different diseases might differ from each other, especially when they are very closely related clinically. So I feel that that's one way, avenue where the field can progress to, like looking at how RNA looks like in all these different subsets. With that, I'd like to thank you for your attention and end my talk here.
Thank you very much, Sam. It was very interesting to hear about this new test that you are presenting and the results. Please write your questions in the Q&A box, as we will go for a final discussion in a panel after all the sessions. So it is my pleasure now to introduce Hanna IJspeert. Sorry, I hope I said your surname perfectly. She's the group leader of Primary Immune Deficiency Disorder Group at the Department of Immunology at the Erasmus Medical Center in Rotterdam, the Netherlands. And her current research line focuses on granulomatosis disease in patients with inborn errors of immunity, and she's responsible for the functional validation of genetic defects. She will today talk to us about phospho-assays for research and diagnostics in inborn errors of immunity. So thank you, Anna. The stage is yours.
Thank you. I hope you can hear me and see my screen. So today I would like to discuss with you the use of phosphoflow as functional readout for genetic variants in EAE. As you know, many EAE variants affect signaling pathways like the JAK-STAT pathway, the PI3 kinase, the MAP kinase, or the NF-kappa B pathway. And actually, I would like to discuss with you, is there place for phosphoflow in the diagnostic work of patients with inborn errors of immunity? But first, what is phosphoflow? Phosphoflow is measuring actually phosphorylated proteins using flow. So when a ligand binds to its receptor, this receptor can get phosphorylated, and this also leads to an activation cascade of downstream proteins, which can also be phosphorylated. And by phosphoflow, you just use an antibody that recognizes the phosphorylated form of this specific protein. And this you can measure with flow cytometry. And today I would like to illustrate the use of this phosphoflow in the diagnostic workup of 3 children with disseminated mycobacterial infections. And as you know, interferon gamma signaling is crucial for the defense against mycobacteria. Here's a simple overview of this signaling cascade between the macrophage and the NK or T cells, in which IL-12 receptor, STAT4 activation leads to the expression of interferon gamma, which can bind to the interferon gamma receptor, which lead to activation of STAT1 and also the phosphorylation of STAT1, and this helps to eradicate this mycobacterium. So when we have had these patients with mycobacterium infections, we expected actually genetic variants in this pathway, because many of you have already shown that genetic defects in this pathway lead to susceptibility for mycobacterial infections. So whole exome sequence analysis was performed with a bit filter of the currently known genes in the according to the IEI's classification. But in all 3 patients, the genetic analysis was not conclusive. So in the first patient, an heterozygous variant was identified in the interferon gamma receptor 1 gene. This was a known pathogenic variant. So that could make sense. Only we expected an autosomal recessive disorder. Actually, there was no second variant on the other allele identified. So then we wondered, is there a true defect in this gene, and should we perform additional molecular analysis? In the second patient, we found two variants. So the patient was compound heterozygous for also two variants in the interferon gamma receptor 1 gene. One variant was pathogenic and the other one was likely pathogenic. But the combination of these two variants was not described before, so we didn't know exactly how severe is the effect on STAT1 signaling. And on the third patient, we identified an heterozygous variant in STAT1, but it was a de novo variant of uncertain significance. So then we needed to know what is the effect of this variant on STAT1 function. So we performed phosphoflow for STAT1 as a functional readout. So we isolated PBMSEs from fresh blood. We rested the cells for an hour at 37 degrees. Then stimulated the cells with interferon gamma or interferon alpha for 5 to 15 minutes. And we use both interferon gamma and interferon alpha because if you look on the left on this pathway, of course the interferon gamma receptor 1 gene is important to form a complex for the interferon gamma receptor. So if you stimulate with interferon gamma, it binds to the interferon gamma receptor, which leads to activation and phosphorylation of STAT1. If you stimulate with interferon alpha binds to a different receptor, the IFNA receptor. It also leads to phosphorylation of STAT1, but in the patient 1 and 2 had a variant in interferon gamma receptor 1, you don't expect any problems in interferon alpha signaling. So therefore we also took that as a control. After stimulation, we fixed the cells, we permeabilized them, and then we stained the cells, and then we measured phosphostat1 and CD14 positive monocytes. And here at the bottom, you see an example of a healthy control. So when you don't stimulate the cells, you see no expression of phosphostat1. If you stimulate the cells for 5 or 15 minutes, you see a nice shift in the expression of phosphorylated STAT1. We performed this assay in all 3 patients. So on the left, you can see the result for the stimulation with interferon gamma, and on the right, you can see it for the stimulation with interferon alpha. And if we look at, so the green bars are the healthy controls, and if you look at the light blue bars, that are the bars for patient 1. If you look at interferon gamma stimulation, we could not observe any phosphorylation of STAT1 in patient 1 after interferon gamma, but normal phosphorylation of STAT1 after interferon alpha stimulation. So this indicated that indeed we only found one variant in these patients, that we had to look further with other molecular techniques, and that's what we did. And then we found a second variant on the other allele, which was an insertion which initially could not be detected using the whole exome sequencing. And because the effect on interferon gamma signaling was so severe, it was decided that this patient would undergo a bone marrow transplantation, which was successful. In patient 2, we were not sure what the effect of these 2 variants was on the signaling. And if you look at the dark blue bars, you see that the phosphorylation of STAT1 in this patient was strongly affected, but we still could see some signaling after stimulation with interferon gamma, and the stimulation upon interferon alpha was normal in these patients. So there was a strong reduction, but there was still activity, so therefore it was decided to go for watchful waiting in this patients. In patient 3, we had this variant of uncertain significance in STAT1. We do see some phosphorylation of STAT1 upon interferon gamma stimulation, but it's significantly reduced. So also in these patients, we decided to go for watchful waiting. So in the previous patients, we saw hypophosphorylation of STAT1, but you can also have hyperphosphorylation of STAT1. For example, in the COPA syndrome, it was described in 2020 by Lapelli et al. That variants in COPA gene can lead to abnormal signaling of STING, which lead to increased interferon expression. And they already showed that in unstimulated cells, you can have an increased expression of phosphostat1. We also had one patient in which we tested, in unstimulated conditions, here in blue you see increased expression of phosphostat1 in the patient, but when you stimulate with interferon alpha, you see a strong increase in phosphorylation of STAT1. So here in this case, in this genetic defect lead to hyperphosphorylation of STAT1. At this moment, the functional assays for STAT1 and STAT3 are currently implemented in our diagnostic laboratory at Erasmus. And I'm currently setting up a functional assay for APDS for routine diagnostics. So as you all know, APDS stands for activated PI3 kinase delta syndrome, which is characterized by an antibody deficiency, lymphoproliferation, autoimmune disease, and they are susceptible to develop malignancies. This disease can be treated with a PI3 kinase delta inhibitor, and it can be caused by either gain of function in the PIK3CD gene or loss of function in the PIK3R1 gene. But both variants lead to hyperactivation of the PI3 kinase pathway. So you have PI3 kinase signaling is downstream of the B cell and the T cell receptor. So you can see in these patients increased phosphorylation of X and increased phosphorylation of S6. So if you look in literature, many publications described increased phosphorylation of phospho-acts in patients with APDS, even in unstimulated cells. We are currently trying to set up a functional assay for this, but in our hands, phospho-acts is not always working so nice. The shifts we see are quite small. So for a functional test for routine diagnostics, we also include phosphorylation of phospho-S6. So our current protocol is that we isolate PBMCs from fresh bloods. We rest the cells overnight at 37 degrees. Then we stimulate the cells with anti-IgM or PMA aminomycin for 5 to 30 minutes. We fix and permeabilize the cells and then stain the cells. And then we measure phospho-act and phospho-S6, and we now mainly focus on B cells. So this is some data from a healthy control. So if you stimulate B cells with anti-IgM, you can see an increase in phospho-acts, but the difference is quite small. If you look at the difference in phospho-S6, you see a larger difference. So upon stimulation of B cells with anti-IgM, you see an increase in phosphorylated S6 protein. So that's still ongoing work, we also encounter quite some difficulties, and I also know that other people have some difficulties. So I think in the remaining minutes, I would like to discuss some of the challenges of phosphoflow, but not before I want to really emphasize that I think that phospho-assays can be a fast functional readout for variants of uncertain significance in the EAE. And I think the power is that it can be applied to all major immunological pathways like the PI3 kinase, the STATs, the NF-κB and the MAP kinase pathways. And I think also with the new developments of spectral flow cytometry, we are also able to combine several phosphoproteins in one assays, because these the expression of these phosphoproteins is usually quite dim. So with normal flow cytometry, it's you want to choose a bright signal, but with spectral flow cytometry, we can combine multiple dim proteins in the same assay. So that's the positive side. I think there are also quite some challenges because phosphoflow is just one of the puzzle pieces which make the complete puzzle for the final diagnosis of a patient with ENE, because not all genetic variants lead to changes in phosphorylation. I showed you the example of the 3 patients with disseminated mycobacterial infections. And the difference in phospho-STAT1 for the patient with the STAT1 variant was not so strong. And also in after interferon alpha stimulation, we did not really observe differences for these patients, while STAT1 is downstream of this pathway. So we also combined this with other assays, for example, the induction of SOX1, which is an STAT1 regulated gene, and then we do observe differences. And I think there are also many more different assays you can do to make this final diagnosis. So additional functional assays might be necessary, complementary to phosphoflow. I think at this moment, phospho-assays are mainly performed in research and not so much implemented in routine diagnostics. And that's because phospho-assays are quite difficult to set up. So each phosphoprotein requires different stimulation times, target cells in which you have to look. And also not all antibodies work well in combination with the harsh buffer you use to stain for these phosphoproteins. Sometimes you see unexplained hypo or hyperphosphorylation, for example, also in healthy controls. And because it's a functional assay, the quality of the cells is crucial. I think also the previous speaker nicely mentioned it, that cells can be affected by transport, temperature, also cell handling. And I think also creating a large database, validate the assays is quite a challenge. So there are no real good standardized protocols available yet. So it's also quite difficult to compare the data between different centers. So actually this presentation is also a shout out to different centers. So can we collaborate to optimize and standardize these assays to make them available for as many patients as possible? Because there are so many variables and factors that it's quite difficult to validate these assays in a single center. So finally, I would like to acknowledge the people in my group, especially Femke and Maaike, who do most of the phospho-assays. The people from the Laboratory of Medical Immunology, our diagnostic lab, including all the medical immunologists, and the clinicians from our pediatric and internal medicine departments. Thank you.
Thank you, Hanna, for this interesting presentation in which you showed nice data and also mentioned the pitfalls and the limitations, and also reach out for further collaboration. I see popping up some reactions in the Q&A. So thank you for now. If there are questions, please type them in the Q&A, and then we answer them after the talk of Evelien. Evelien Sprenkeler is medical immunologist in training at the Radboud University Medical Center in Nijmegen, the Netherlands. And she did her PhD under mentorship of Professor Taco Kuipers and Dr. Robin van Bruggen in Amsterdam, where she focused on neutrophil host defense mechanisms. And today she will talk about disorders of neutrophil development and function.
Yes, thank you, Mirjam, for the introduction. Today I would like to talk about neutrophils. First, a small introduction about them. So they're also called polymorphonuclear cells because they have the multilobulated nucleus and they're part of the granulocytes. They're the most abundant innate immune cells, and 50 to 70% of the circulating leukocytes are neutrophils. They mature in a bone marrow, which takes about 2 weeks, and they're produced in massive amounts every day. It's estimated that this is around 100 billion per day, but this is also necessary as they're short-lived cells, and it's estimated they only live 1 to 2 days in the circulation. So neutrophils are important effector cells in a host defense against invading microorganisms, specifically bacteria and fungi, and the first immune cells recruited to the site of infection. So once they arrive at the site of infection and they recognize the pathogen, there are several host defense mechanisms to try to kill these pathogens, which include phagocytosis, degranulation, the production of reactive oxygen species, and the formation of neutrophil extracellular traps. So in the last years, increasing amounts of genes which are associated with inborn errors of immunity have been found. And if you divide them in subgroups, 41 genes are associated with phagocyte number and function. And these genes are associated with congenital neutropenia, defects of motility, defects of respiratory burst, or other non-lymphoid defects. So today I would like to tell you something about functional assays that exist to investigate neutrophil function. And I will start off with functional assays to detect neutrophil motility defects. And I will show a combination of flow cytometry assays as well as microscopy. So I will tell you something about neutrophil motility defects by describing leukocyte adhesion deficiency and MKO1 deficiency. These are two inborn errors of immunity. So as the name already says, leukocyte adhesion deficiency means that neutrophils can't adhere. And this means specifically to the endothelial cells which line the blood vessel. And because neutrophils are unable to adhere to the endothelial cells, they're not able to transmigrate into the tissue. So they can't exit the bloodstream and enter the tissue to kill microorganisms. So patients with LAD suffer from recurrent bacterial infections, leukocytosis, because the neutrophils can't leave the bloodstream. Characteristic is also a delayed separation of the umbilical cord and impaired wound healing. So there are three types of LAD, LAD 1, 2, and 3. And in LAD 2, the neutrophils, they have a defect in rolling because they have a defective fucosylation of selectin ligands. And for leukocyte adhesion deficiency 1 and 3, there's a defect in beta-2 integrins. In LAD 3, the beta-2 integrins, the inside-out signaling is defective, leading to impaired integrin activation. And for LAD 1, there's an absent or decreased beta-2 integrin expression caused by mutations in the integrin beta-2 gene. So these neutrophils can't firmly adhere to the endothelial layer and can't transmigrate. So the expression of beta-2 integrins can be easily evaluated with flow cytometry. So here you have the histograms for the staining with CD18, which is the beta-2 integrin. So in the control, all neutrophils express beta-2 integrin. And here in the patient's condition, so these patients have leukocyte adhesion deficiency, no beta-2 integrin is expressed on the neutrophils. I would like to switch to MKO1 deficiency, which is fairly newly reported inborn error of immunity. MKO1 deficiency stands for megakaryoblast leukemia 1, and it's a transcriptional co-activator imported for actin regulatory gene transcription. In the cytoplasm, MKO1 is bound to globular actin, also known as G-actin, and when there's cellular activation, G-actin is incorporated into the F-actin strand, also known as actin polymerization, and this will lead to the liberation of MKO1. MKO1 can then translocate to the nucleus, where it can bind to serum response factor, and this transcription factor is essential for cytoskeletal target gene transcription, including actin itself. So to date, only 3 patients with MKO1 deficiency have been described, and these patients suffered from severe infections, which were potentially fatal. So these patients, they have a problem with actin polymerization, and you can assess this by flow cytometry and using a specific dye for F-actin, which is phalloidin. So here neutrophils have been isolated and stained with phalloidin. And what you can do is you can stimulate your neutrophils. So in black, you stimulate control neutrophils with FMLP, so a bacterial chemotactic factor, or complement component 5A, which is also chemotactic. And then immediately, already after 10 seconds, you see a massive increase in filamentous actin fluorescence. And this slowly goes down again, indicating depolymerization. So in a control situation, you see this nice increase. And in a patient, the MKO1 deficient patient, you can already see that the filamentous actin levels are at basal state already lower than the control. and not much is happening when you stimulate these neutrophils. You can also look at filamentous actin by microscopy. So here in magenta, you see the filamentous actin nicely expressed upon stimulation in the control condition, but not in the patient condition. So because the actin polymerization is not working correctly, we also wanted to look at the chemotactic response, and you can do this via transwell assay. So chemotaxis is the directed movement of neutrophils towards the site of infection, and it's essential for the extravasation of neutrophils from the blood vessel. So in this assay, neutrophils need to transmigrate through 3 micron pore size filters, and they are going towards several chemoattractant factors. And as can clearly be seen in the patient condition, they are lagging behind. We can also visualize this with a TAXI-scan analysis. So here neutrophils are moving towards a chemoattractant on the top side. And you can see in the control condition, this is nicely and efficiently been done. However, the patient neutrophils are also not efficiently going towards the other side. Next to that neutrophils need to get to out of the bloodstream into the tissue. Once they're in the tissue, they of course need to kill the pathogen they encounter. And one of the ways they do that is by producing reactive oxygen species. And I want to talk about functional assays to detect the neutrophil respiratory burst. And you do this mainly by for the identification of chronic granulomatous disease. So what is the respiratory burst? So upon cellular activation, the NADPH oxidase enzyme complex is formed, which is composed of membrane-associated subunits, which is the GP91-FOX and the P22-FOX. And you have cytosolic components, P40-FOX, P47-FOX, and P67-FOX. So once the complex is assembled, which can be on a phagosomal membrane or on a plasma membrane for extracellular ROS production, oxygen is catalyzed into superoxide production. And it doesn't stop there. So the superoxide can also be catalyzed into hydrogen peroxide by superoxide dismutase, and also MPO, myeloperoxidase, which is derived from the azurophilic granule, can catalyze hydrogen peroxide again in hypochlorous acid. And these factors are highly antimicrobial. So this helps for the killing of pathogens. So chronic granulomatous disease is a very rare primary immunodeficiency, and it's caused by autosomal recessive or X-linked mutations in one of the genes of the NADPH oxidase complex. So if you have mutations in CYBB, which makes GP91-FOX, this is the X-linked form of CGG, and this is also the most common form. 65% of the patients with CGG have this form, and males are the only ones affected, of course. And the other subunits, they are caused by autosomal recessive mutations. So because the NADPH oxidase complex is not functioning, there's an impaired production of reactive oxygen species by phagocytes. And this leads to recurrent and severe bacterial and fungal infections, especially from Staphylococcus aureus and Aspergillus fumigatus. So how can we assess the respiratory bursts? We can do so by using dihydrorhodamine 1,2,3. So this compound is non-fluorescent. However, when there are reactive oxygen species produced by neutrophils, so for example, upon stimulation with PMA, DHR 1,2,3 is reduced into rhodamine 1,2,3, and this is fluorescent. And we can measure this fluorescence by flow cytometry. So here you will see histograms, how it looks in healthy control. So left the unstimulated condition of the neutrophils, and right the PMA stimulated condition of the neutrophils. So you see a nice shift in the fluorescence. However, when you have an X-linked CGG patient, you see that there's no fluorescence of DHR in the neutrophils. And for the carrier condition, so for example, the mother of this child, you can see that this mother has a fraction of her neutrophils do produce reactive oxygen species, and a fraction of the neutrophils don't. And also the recessive disorders also show defective reactive oxygen species production. However, it's important, if you see an abnormal DHR response, you need to check if there's not an MPO deficiency, because MPO deficiency can also lead to a strongly decreased DHR signal. And you can do this, for example, by flow cytometry within the anti-MPO antibody. So apart from the respiratory burst assessment, the functional assessment, you can also look into the subunits with flow cytometry. So you can do this with flow cytometry. You can detect all the different subunits. So here, GP91 expression, P22 expression, P47 expression, and P67 expression. They're all present in the healthy control. However, in the patients with an X-linked GP91 deficiency, you can see that the GP91 expression is absent, and also the P22 expression is absent. And it has been described that the membrane-bound subunits need each other for expression. So if one of them is not there, the other one will also not be expressed. And indeed, the other way around, the P22 deficiency also doesn't have the GP91 FOX expression. And there are also assays to detect neutrophil development defects. So the development of neutrophils starts in the bone marrow until they fully mature and they enter the bloodstream. And I will illustrate this by illustrating this by a GC-CSF receptor deficiency. So a lot of inborn errors of immunity are caused by congenital neutropenia. And as I said, the neutrophil matures in the bone marrow, and you can nicely investigate neutrophil maturation by flow cytometry for CD11b and CD16. So starting from a promyelocyte into a metamyelocyte, bad neutrophil, and then a segmented neutrophil, fully mature neutrophil, there's an increase in expression of CD11b and CD16. And in blood, you should only have mature neutrophils, which are CD16 and CD11b positive. So there are several congenital neutropenias and genes described which cause them. And I will focus shortly on CSF3R, which is the GCSF receptor. So there are lots of function mutations described in the GCSF receptor. And GCSF is a growth factor, which is a key regulator for neutrophil development and the proliferation and differentiation of myeloid progenitors. And it plays also an important role in the emergency granuloparesis in case of infection. So in these loss of function mutations, there are mutations in the extracellular domain of the GCSF receptor, so there's no signaling. And with flow cytometry, you can nicely look at the expression of the GCSF receptor on neutrophils. So in the left, we have a patient from the UK, where we stained for the GCSF receptor. And you can see that compared to the control, there's a very much decreased expression of GCSF receptor on the surface. And also for a family in the Netherlands, we have two patients who have homozygous missense mutations in the GCSF receptor, which lack expression on their surface. However, if you then look at the control and the unaffected CYP, it's nicely expressed. And interestingly, the heterozygous family members show partially reduced GCSF receptor expression. So I want to conclude that functional studies increase our knowledge on neutrophil host defense mechanisms. And I truly believe that if you better understand the biology behind the disease, the clinical management regarding patients with these rare congenital disorders can be supported. And as I illustrated, there are numerous of functional and flow cytometric tests available to investigate neutrophil function. I didn't mention all tests, of course. You can also look at apoptosis, degranulation, phagocytosis. However, it has to be mentioned that these are very rare diseases, and functional studies on neutrophils can be challenging to the nature of the cell. So they are very short-lived, they're easily activated. So I think there's an important role for the expertise centers and laboratories if you really want to dive deep in neutrophil function. And for future perspective, as I said, more genetic variants are identified. So I think there also needs to be more needs for functional studies. And with this, I want to conclude my presentation. And for at least in the Netherlands, Sanquin in Amsterdam is really the expertise center for neutrophil studies. So if you have any interesting patients, I would recommend to ask them to think along. Thank you.
Thank you very much to all the speakers for the really exciting and excellent talks we heard. I think that was really great. And I think looking at the time, I think we go straight into questions. And Sam, you got tons of questions and you were very busy answering them already. And basically saying, yes, the NK cell assays, degranulation works also when they're not active. And I think that's important to know. And that's why it's an excellent test to screen. You showed also, answered a lot of questions about LRBA, and you're really exciting finding how you can use that CTLA-4 expression intra and extracellularly in this, and that you even have a quite sensitive assay in your hands, at least, to find even maybe not full-blown lack of function. The two questions I would like to point towards you is, one, you were asked whether stimulation with anti-CD3 or anti-CD16, compare that to PMA, and you said you would prefer the stimulation anti-CD3, CD16 because it's more cell-specific. And my challenge for you now is, you can obviously gate back on the cells which responds, so you could look which cell is responding if you stimulate with PMA. And the question is, PMA overcomes signaling defects of CD3, right? So you get a mixed answer. If you don't see a response, it could be the reason you expected this, or it could be that the signaling upstream of PMA stimulation might be defective. And so just a short answer, just looking at the time.
Yeah, so yeah, that was one reason why we didn't want. We wanted the stimulation to actually go through the pathway that it's normally would go through when you're stimulating from the CD3 receptor, PI3 kinase, and the calcium flux, influx, etc. We wanted to see that. We wanted the process to go through the entire system and end up with the degranulation. The other thing that I didn't like about PMA is that it's really, really strong. And we see that sometimes cells don't survive so well after such a strong, overwhelming stimulation. We want something more physiological when you stimulate it. So that's the other reason why we didn't go with that. We did try.
Yeah, perfect. Second question is the RNA signatures. So you were asked, what are they good for? And you said they're good for everything. But we need to have better data on that. And I absolutely agree with you. I think there is a future in there. Could you just make us a bit more careful about the hope we have with RNA signatures and potential downfalls on RNA signatures, and where do we get them, at what time do we get them, and what cell, and so on?
Yeah, so when the regular way of getting RNAs is through PAX gene tubes or Tempest tubes, and the downside of that is that you're getting all the cell types. You're getting a signature of all the blood cells at one point. But the good thing about that is that the Tempest or the PAX gene tubes, they are very stable. You can freeze them. They are you can send them room temperature for 3 days. They are stable, or you can freeze them and they're stable for like 8 or 11 years or something like that, according to Kaigen. That's the good point. Like you can get faraway samples. You can very easily freeze and collect samples for laboratory work. The bad thing is you are getting the entire, all the signals from all the cell types. But if you want to get individual signals, like you need still a lab to isolate B cells or T cells or whatever cell type that you want. And that's very laborious. Number one, that's more time consuming. And you get the same problems with shipping. You want cells that are alive and not activated due to like too much heat or poor transportation, etc. You come with the same problems. And when you isolate out T cells or B cells, you end up with very few cells. And therefore the RNAs, the signal is much weaker compared to whole blood when you immediately collect it in PAX gene tube. In terms of the results itself, you first have like a training set of samples where you just screen through them and find a signature, and then you have another population where you look to see if it actually works. The thing with that is you need to have very clear clinical definition of your cohort, because one center might define it a different way and another center, and when you then compare signatures, it might not compare well against. This part of the research is at its infancy right now for IEIs. There's still a lot more to be done. We haven't really started with it. So everything that we say right now is really postulation.
Yeah, no, thank you very much. And I think it will not only the cohort, it's the individual who is sick or not sick, as we said before, and that will strongly affect, obviously, signatures in this different population. And need to know the composition of your blood populations to have an interpretation which is valid in there. So thank you very much for that. And I hand over to Clara.
Thank you. So we have also some questions for Hanna. I'll try to summarize them. And in regards of the PIK3CD variants, they are asking if it would be enough to validate these variants by measuring only the PS6 and not others like PIKT, or if you definitely have to do both tests. And related to this question, it would also be of interest of the audience to know, how do you manage to do the test with so little numbers of B cells in the patients? Thank you.
Yes, thank you. The test for APDS, we're still setting this up. So I definitely agree with that APDS patients have really low numbers of B cells. At this point, we are still struggling a little bit. How should we measure the T cells? The T cell stimulation, I'm not so happy with it yet. You're still also, for example, puzzling, should we do it in T cell blast, or should we do it now on the T cells as we do it now with overnight culture? So I think I would definitely recommend also to look in T cells, but I think at this moment we are still a little bit struggling how to optimize this assay in T cells. So that's, I think, still a work in progress. And I also saw many responses on people that want to share also protocols. So I think that's really great. So I hope also that with the community we can also share experience in that kind of perspective, because in that side we are struggling a little bit with that. And I have to think about, is S6 enough? S6 is downstream in the pathway, but also downstream other pathways. So I definitely think that S6 alone is not enough. So I would definitely love to combine it with, for example, phospho-assays. And we now use the serine 473, but I also know that actually the other at the 308 phosphorylation site is actually nicer, but also there we still have to optimize the assays. So I would definitely recommend to combine ACT and S6, because S6 is much more downstream in the pathway and also activated by other pathways.
Thank you, Hanna. In the matter of time, we'll move to the next speaker's questions, but just to make you know that the speakers will get the questions, so they will be able to answer privately to each of you. Okay, so nothing will be remained unanswered.
Okay, that's good. There was a question to Evelien from, do any of the gene mutations causing CGD have normal DHR?
That's quite interesting. For the P40FOX mutations, it has been described that DHR can be normal if you stimulate your cells only with PMA. So you also need to have a particulate stimulation like E. coli included in the assay. Yeah, that has been described, but it's very rare. Yeah.
Okay, and there's another question that regards the normal value for CD18. Are there any patients, especially adult patients, with low CD18 and poor wound healing, mucosal infections, etc.?
I don't know if CD18 expression is, for leukocyte adhesion deficient type 1, you have a range of how much CD18 expression is still there. So if you do severe form, then it's less than 1%. And I think if you have moderate, it's up to 30% expression of CD18 still there. So there is indeed a range, but you can also do a functional assays, for example, to see how many neutrophils are able to adhere still.
Okay.
Yeah.
Thank you, Evelien. We are come at the end of this grand round. I think it was a really great presentation, a lot of information. And I also would like to indicate that Clara, she's chair of the educational working party. She's trying to collect also the protocols for various functional assays. And there's also an initiative from the genetics working party to make big list of functional assays, whether they're available in which laboratory, because I think especially the assays that has been presented and discussed today will not be possible to run in every lab. So I think we need each other to develop in the end the best protocols. But I think it's also good to focus, because not everyone can do everything, and therefore also we need each other. And I think ESID is a good society to share the experience and the knowledge. I would like to thank for joining, and I hope to have the next grand round also very interesting. Thank you very much.